Good morning, everyone. I'm Anne, and I have a history in antibodies and antibody discovery, working with discovery and development of antibodies in industry in the field of cancer during the last 20 years. And then I come across this neglected disease and snake bites. And I didn't know very much about it and was kind of shocked about the big need for better treatment in this field, and got this feeling maybe there is a way to bridge my knowledge in antibodies into the field of snake bites. Is that something I can do to actually help those victims? And luckily, there is a bridge. There is a bridge between Sweden, where I live, and Denmark, where we have the Center for Antibody Technologies, led by Andreas Lausen. So I joined his group about a year ago, trying to bring my knowledge in antibodies into snake bites. And that's what I'm also here to learn about today. So I will talk about recombinant oligoclonal broadly neutralizing antibodies and how we can use them for snake bites. And I don't need to convince this audience that snake bite causes millions of it affects a lot of people yearly. Let's just put it that way. It's not only the people dying, it's also the social impact this has. And in my project, the work we're doing, we focus on sub-Saharan Africa. So luckily, there are traditional antivenoms. You know, you immunize large animals such as horses, they develop an immune response, you purify the antibodies, and they're given as antivenoms. These products has some drawback. The horse doesn't respond the same way each time. You get different immune responses. Horses are different, so there are batch-to-batch variations. It's very expensive. Since it's not a human product, it can cause really adverse reactions. It's a low therapeutic content, and they are expensive. So even if they're super good, is there any way where we can improve this? Maybe make it better? We think so, by doing a recombinant antivenom. If we have the genes for the antibodies, they can be produced in CHO cells in large scales, and in this way, we can produce the antibodies. But we do need the genes for these antibodies. What else do we need if we want to do an affordable recombinant antivenom? We don't know everything, but some things that we can imagine is that we do need the genes to be able to do this. We need to have antibodies of high affinity. The antibodies need to be broadly neutralizing. Maybe we also need different antibody formats. So how can we do this? Well, we start with the genes. I worked a lot with phage display technology, and this is also what we do in our group. And phage display is a way to link the genotype and the phenotype of an antibody. So you have the antibody on the outside of the phage, the protein, you can access how good is this antibody. Do it bind to the toxin we're interested in? Inside the phage, you have the DNA, you have the code, you have the map. How do I do this antibody? If you then have a really huge library, it's like when looking for a book. If you have a huge library, it's likely that you can find the book you're looking for in there. In this case, it's an antibody. So we have a human naive single chain library consisting of more than 40 billion different antibodies. We also have immunized llamas and have an immunized library, which I will come back to. But we're using the phage display technology, which is the same in both cases. So it starts with presenting your antigen, the toxin, and you add your library, which means that all antibodies that are able to bind to the toxin of interest will bind. You can wash away all the antibodies we're not interested in. Off they go, we only have the binders left. These can be eluted. We then have the DNA, how to do these antibodies. They can be amplified in bacteria, and then you can repeat this circle, typically 2 to 4 rounds. We then screen individual antibodies, confirming do they bind to the toxin, and sequence them to see that they are different. And then we can evaluate, do they neutralize our venoms in vitro and in vivo? So starting with one snake, the black mamba, and the dendrotoxins in this, we did phage display, found some antibodies, and evaluated them in vivo, mixing antibody, toxin, injecting into the mice and looking at survival. We can see to the upper left in red, if we inject only toxin, mice die. If we add one antibody in blue, yeah, some mice survives, but not all of them. If we instead take 2 antibodies, further to the right in blue, half of the mice survive, but it's still not all of them. If you take another mixture of antibodies, it's still survival of half of the mice. But in the lower graph, if we have a mixture of 3 antibodies binding to different dendrotoxins, all mice survive. Save the mice from the dendrotoxins. It's also dose dependent. So if we have a 3-to-1 molar ratio between the antibody and the toxin, we have full survival. If we have less antibody, some of the mice dies. Okay, so this shows that this can be done. We have the black mamba, we have the dendrotoxins, we can neutralize this. But as you all know, snake venoms are very complex. And I said I'm working with sub-Saharan Africa. There are very many different snakes in this area. Actually, if we look at the 26 most medically relevant snakes, according to the WHO, and look into the venom composition of these, it contains more than 300 different toxins. So we need to neutralize more than 300 toxins to have a treatment for all of these snakes. That means we need to have as few antibodies as possible doing all this work. And one thing that is good for this is to have antibodies of high affinity. You can have one antibody, one toxin, and if you can go to that low ratio, that's as few antibodies that you can get. So is there a way to improve the affinity if we find an antibody that is not good enough? For example, from our naive library? Yes, you can use a technique called light chain shuffling. If we take the single chain, that's the binding part of an antibody, the variable heavy and variable light chain. Normally, the heavy chain is the one most responsible for binding. And then we can take very many different light chains and see, can we improve the affinity by changing the light chain? In this case, we did that, found a new antibody, converted back to IgG, and look, does this better affinity also give better neutralization? Because that's what we're after in the end. So here we have an antibody binding to alpha cobra toxin and looking at the inhibition of alpha cobra toxin binding to the acetylcholine receptor. And you can see in black, the original antibody dose response, we do inhibit alpha cobra toxin from binding. But if we have an affinity approved antibody in blue, we can use lower doses, less antibody, and still inhibit the binding. So we can improve the affinity. Can we also make broadly neutralizing antibodies? And why is it important to have broadly neutralizing antibodies? I said we have 300 toxins approximate that we're targeting. If we look at an example over here with 3 snakes and 3 toxins, if we have 1 antibody against each toxin, it's 9 antibodies. If we can find 1 antibody that recognizes the same toxin class but from multiple species, we reduce the number of antibodies needed. In the example, 3 antibodies instead of 9. Imagine the same mathematics on 300 toxins, it's less antibodies. Good. A way to achieve this during the phage display selections is something we called cross-panning. So while you take your antibody library and expose it to the toxin, you use 1 toxin in the first round, finding all antibodies capable of binding to this toxin. Then you take the output and show a second antigen coming from another species. And then you only have antibodies left that are able to recognize both of these toxins. And then you can add a third one and so on. So does this work? Yes. We did cross-panning and found antibodies against alpha neurotoxin. And this show a pre-incubation assay where we have one antibody binding alpha neurotoxins, and then we'll look at the survival of mice. So pre-incubation assay, having the toxin, having the antibody, inject it, and look at the mice survival. If we take whole venom from Nyakotia, all mice survive. If we take whole venom from the king cobra, we still have a protection. More mice survive. We prolong the life of the mice. They die eventually. If we take the whole venom from black mamba, also here we see a protection, and then they die. And this that we don't see 100% protection in the black mamba and king cobra is because likely their venom contains not that much alpha neurotoxin compared to Nyakotia, going from 55% down to 13% in the black mamba. Then we get this question, from reviewers and so on. Well, a pre-incubation assay is not exactly how snakebite happens in reality. So we did a rescue assay, injecting whole venom from Nyakotia subcutaneously in the mice, and then giving the antibody. And if we give the antibody intravenously directly, we save the mice. So also in a rescue experiment, we with one antibody totally rescue mice injected with whole venom from Nyakotia. So the antibodies are broadly neutralizing. They are of high affinity. Do you think we also need antibodies of different formats? Maybe. And one format that has gained increasing interest during the last year are nanobodies. They are derived from the heavy chain only antibodies that are found in cameloid animals, such as llamas. And you take the binding part, the upper part in blue, and that binds as a normal antibody with high affinity, high specificity, but it's much smaller than a normal full-length IgG, which means that it's cheaper to produce. It's easy to reformat in different ways. It has high stability, high solubility. It's also good at tissue penetration. So we try to take the best of the in vivo and the in vitro world, immunizing llamas with whole venom from either 8 viperids or 18 elapids in a dose escalating way. So the llamas mount antibodies towards these venoms. And then we made phage display libraries from those animals and use phage display to fish out the best antibodies from these llamas. And this is one example where we have nanobodies towards PLA2s in elapids. It's an in vitro assay, so we just look at the PLA2 activity. And in gray, we see the activity of whole venom from some different Nia species. And in black, we see different nanobodies that are able to inhibit the enzymatic activity from all these different Nia species, which means that we can find broadly neutralizing antibodies and nanobodies towards multiple species. So where are we in this whole process of finding an oligoclonal cocktail for sub-Saharan Africa? As I said, we have the 26 snakes, and we have looked into the complexity of the snake venoms, identified the 300 toxins that we need to neutralize. We have multiple molecules, both antibodies and nanobodies, towards some of these toxins at different stages in our pipeline. We're currently producing the first batch of oligoclonal mixture, looking into this show cell production where we produce more than one antibody at a time. So with that, I'd like to thank all the fantastic people within the Center of Antibody Technologies, especially Andreas Lausen and Jessica, Jiang, Carol, Sherin, and Alex, Tasha, and Camilla. And I'd like to thank all our great collaborators and our sponsors giving us money, making it possible to do this. And I'd like to thank all of you for your attention, and happy to take any questions.
Julian? Hi, thank you. My name is Julien Potet from the MSF Access Campaign. Thank you for your interesting presentation. I had a question about the strategy itself. Have you considered possibly to some sort of a hybrid solution, like providing monoclonal antibodies or nanobodies on top of existing antivenoms for toxins that are not very well neutralized by the existing antivenoms? I'm thinking for sub-Saharan Africa, we know that existing hyperimmune equine antivenoms are not very effective against cobra neurotoxins. So maybe an intermediate solution would be to provide these antibodies to existing manufacturers so that they could put it into the final product. Is that an intermediate solution?
Of course, there's possibility to improve and fortify the current antivenoms also. And that's also something we're looking into. And I think it will be, we will not have a complete recombinant antivenom tomorrow. And that is probably the way to go to start adding antibodies to the current one and then maybe shift towards to others. But it's definitely yes.
David?
Yeah, I've just got, I guess, a practical question. Imagine you are able to commercialize this.
Yes.
What do you think the likely cost of production of a broadly neutralizing recombinant antivenom is likely to come to?
Yeah, I don't have the figures, but it's the R&D discovery costs that are high, and then the production costs will go down. Looking at, for example, nanobodies are today added as food additives and so on, so they can be produced really cheap. And I think costs also for full-length antibodies are going down, but I can't give you a number.
Yeah,
but it's an important question, of course, that it's affordable.
It's a real concern for in the one place where snakebite is a really huge problem, and that's India.
Yep.
Because the price of antivenom is fixed by the National Pharmaceutical Pricing Authority, and they will only pay you around about 436 rupees per vial, and a conventional antivenom is about 500 milligrams per vial. So for half a gram, you're going to get paid less than €5, and they are completely intractable. This is why manufacturers of conventional products are actually losing money at the moment, because they're being paid less than it costs them to produce the product. And I think there has to be a strategy to overcome that if we're going to see these sorts of products actually reach the point of market penetration. So I think it's a really important thing that in the R&D pipeline, you have to consider how much is it going to cost, and how are you going to deal with the issues of getting it into markets where there are very strict cost constraints.
Yes, I agree. You have to take the whole chain into consideration, of course.
Jan Titkat, and then I will ask also my last question.
Thank you for the very nice presentation. In terms of neutralizing effect, and you compare nanobodies with the classical monoclonal antibodies, can you say something about the molar ratio that is needed? Does that stay the same? Because with nanobodies, you may imagine that you need a higher molar ratio to get the same effectiveness.
Yeah, so the lower, we can go to 1-to-1 with both antibodies and antibodies, and then calculating that an IgG has 2 binding sites. So they're equally potent in what we see. Yeah.
And my question is related to this previous one. What's the potency of the products? The potency of the antivenoms that you have told us and the nanobodies? What's the potency in terms of milligram of venom neutralized by gram of product or something like this? Because if you just take a small amount of the most active molecules and you neutralize these, then you neutralize the venom, but in this system, but not in a real bite where you will have all the venom, which all the molecules have a threshold of lethal activity.
Yeah, but remember the slides I showed with this one antibody, it's with whole venom. So it's not only the one.
But how many LD50s? 2?
Yeah, but it's still a game of numbers. So how many molecules do you have of your toxin, even if it's in the whole venom, and how many molecules do you have of your antibody? And that is the same for current antivenoms, the number of molecules you have.
This is why I wanted to compare the figures that we have available for normal or conventional antivenoms with the potency figure of these oligoclonal mixtures.
Yep, yep. But I can't see why it should be higher, since it's still number of molecules per number of toxins. So if you take the milligrams you have in a conventional antivenom, we should be able to have less milligrams of the recombinant ones, since it's all active ingredients.
Well, later I will show you data that says the contrary. But
yeah.
Okay. If there are more questions, then we move. Thank you again.
Thank you.